In this video, we will explore TCGA data using GeneTegra in order to create a single query where clinical patient information and dataset identifiers can be retrieved. The TCGA data model was designed based on the ontology for biomedical investigations. The structure of the OBI begins with the root level node, entity. Everything else subclasses from that point. On the next level are the nodes continuant, which refers to objects and spatial regions, and occurrent, which refers to processes spanning time. For this use case, all of the terms fall under the continuant node. This has three subclasses: independent continuant, which bears quality and cannot inhere anything; generically dependent continuant, which depends on other entities; and specifically dependent continuant, which inheres from other specific entities which cannot change. In the design of the queries for this use case, the displayed independent continuant entities and generically dependent continuant entities were used. To start, a clinical query was created from the TCGA data model. Beginning with the patient entity, connections to the stage event and the clinical stage entities were made, allowing for the value of the stage to be returned. Next, the age of the patient at the time of initial diagnosis is selected. Also included is the vital status of the patient, the days since death, and the days since the last follow-up visit. These last three data points are set to be returned optionally, so the retrieval of the other data points is not dependent on their existence. Executing the query produces a set of results with 582 records. The second query is designed to retrieve dataset information and identifiers for the patient and begins by selecting the patient barcode. Next, the query is expanded through the sample, portion, and analyte entities to retrieve the analyte type. Finally, the analyte information is connected to the aliquot in order to return the aliquot barcode and data type for the aliquot dataset. Executing this query produces a large number of results, which was restricted to a maximum of 5,000 returned records for this run. GeneTegra allows for a new query to be constructed by using previously designed queries. The two queries for this use case can be combined using the patient class as the integrating point in order to return all of the data in a single result set. Upon executing the query, the results can be downloaded and used to retrieve the complete relevant TCGA data sets for the patient. This query was limited to return 10,000 records as the result set. When designing a query, data points can be set as parameters. These parameters can be used by others as points of data restriction when executing the query in order to tailor the results to their needs without needing to modify the query design. Additionally, these parameterized queries can be accessed using the GeneTegra web client, bypassing the need to connect to a GeneTegra client to retrieve results. GeneTegra also includes other valuable functionality. Data that has been retrieved can be saved to a local file as delimiter separated values or as an Excel spreadsheet. Data can also be saved to the GeneTegra server. Queries that have been designed can be saved locally or can be published to the GeneTegra server, where other users can execute or edit them as long as they have the proper credentials. Also, favorite queries can be bookmarked for quick and easy access from the client interface. Multiple data sources of differing types can be accessed and queried against. Additionally, data sources can be integrated and queried in a single instance.